Other than the three main RNA molecules like ribosomal transfer and messenger RNAs, there are several non-coding RNAs. So those three main RNAs, they play a role in transcription and translation. Their role is quite clear. Um, but there are so many other non-coding RNA molecules that also play a very important role in post-transcriptional regulation of the gene expression. They determine after the transcripts are synthesized whether those transcripts need to be translated or not. So they can directly interfere with those RNA molecules and do not either they degrade them or they bind with them in such a way to break them down or do not allow them to be translated. Those RNA molecules are called as non-coding RNAs or ncRNAs or small non-coding RNAs. Non-coding RNAs, they are involved in the interference of RNA, which is called as RNA interference or RNAi. And there are two main types of those RNA molecules, and those are the ones which are called as small interfering RNA, siRNA, or microRNAs or miRNA molecules. So let's talk a little bit more about them. What is their main mechanism of controlling the gene expression? RNA interference is based on binding of some RNA molecules with messenger RNA molecules. So these interfering RNA are synthesized specifically to block the translation or gene expression of messenger RNA, because those sequences that are included in those non-coding RNAs are complementary to messenger RNA. So as if they are really targeting directly to messenger RNA, they will bind to messenger RNA and they will block the translation of those messenger RNAs. Both of these molecules, non-coding RNA molecules, either siRNA or miRNAs, both of those are very short sequences. So both of those are fairly short sequences in length. They are both double-stranded. They are about 20 to 31 nucleotides long. And they come from different sources, and they have very similar mechanism of interference, of binding to messenger RNA and blocking the translation of those transcripts. So that is one way of controlling the gene expression. Small interfering RNAs are slightly longer than microRNAs, but otherwise their activity is somewhat similar. Both of those involve some enzyme complexes like disers, and then they associate with another complex of RISC and argonaute protein. So, but their mechanism is the same. So let's go to the next slide and take a look at the figure 16.12. Both of these mechanisms are shown here in this image. For small interfering RNA pathway, which is shown here on the left side. So this is the small interfering RNA pathway, and on the right side is the microRNA pathway. So let's take a look at the small interfering RNA first. So small interfering RNA is produced inside the nucleus, like any other RNA molecules are produced. In case of eukaryotes, then it is exported to the cytoplasm. Inside the cytoplasm, double-stranded RNA binds with a with an enzyme called dicer. So the dicer enzyme will break it down into smaller. So it is broken down. It binds with the dicer, and it breaks it down into smaller fragments of double-stranded DNA, a double-stranded RNA molecule, which is shown here in the second step here. Small interfering RNA is produced from a longer double-stranded RNA molecule. Now, this one, after it breaks down, and then it binds with another protein called RISC protein, and that RISC protein is a protein that binds to that small interfering RNA. So RISC protein is RNA-induced silencing complex. That's what it is. It stands for. So it binds to that small interfering RNA, and then it unwinds it. It becomes single-stranded. So the double-stranded, which we see here, unwinds, and only one fragment of that is left with the RISC and argonaute. AGO is another protein which is a part of this RISC complex called argonaute, or AGO. So it becomes more like a small protein complex that is carrying a single-stranded RNA, which is very small, small fragment of RNA. So that is about 30, somewhere between 20 and 30 nucleotides long. Once that RISC complex with the small single-stranded small interfering RNA, and it finds a complementarity with the messenger RNA, it binds to it and it cleaves it. When it cleaves it, then it is no longer able to produce the protein. So translation is interfered because the transcript is gone, so there is no translation that can take place. So this mechanism may be useful for some of the organisms. For example, in case of some transposable elements or some viruses that attack viruses that may be found inside the cells. The other pathway, which is also very similar, and there are some overlaps between these two pathways of the microRNA molecules, those are produced just like any other RNA molecules are produced in the nucleus, then they're transported to the cytoplasm. In the cytoplasm, they bind with the protein called dicer, the same thing that we have just seen before. So dicer will chop it up into smaller pieces, and those smaller pieces will bind to the RISC and argonaute protein complex. It denatures, it unwinds, only one fragment is left. So that one fragment that is left with RISC and argonaute protein, whenever they find complementarity with messenger RNA, so there could be two things that can happen. If the complementarity is perfect, then messenger RNA will be broken down, so which is shown here with this arrow, meaning that the fate is the same. So with the perfect homology, perfect complementarity between the fragment of RNA which is left with dicer and argonaute protein, then the degradation of messenger RNA will take place. If the complementarity is partial, then what will happen is, then it will bind to it, and it will inhibit the use of that messenger RNA in translation, because now there's a bulky molecule that is attached to it, so it cannot go through the process. The end result is the same, whether it goes to complete degradation or it binds to it and stop it from being involved in the translation process. So that's how they are able to control, even after it is produced, and it is able to control whether these will be able to take part in translation or not. So there's more detail, talks about what these proteins are and how they actually bind to the small fragments, how they produce small fragments that they carry with them so that they can be utilized at the time of of binding to those with the messenger RNAs. There is another protein here that is called Rasha protein, which is shown here in green color. Let me grab my pen. So that protein is shown. Oops. That protein is shown here. This one right here. The role of this protein inside the nucleus is to remove these fragments of toward the 3' end of RNA molecule, which are not complementary sequences. So they are in the form of kind of overhangs on both ends. So this protein trims them from both sides. And once it is trimmed, then it produces this kind of a microRNA, which is transported to the cytoplasm, and the rest of the process takes place on that microRNA there. Okay. MicroRNAs have been found in animals and plants both, and they are encoded by some viruses. And there are at least about 1,500 to up to about 5,000 microRNA genes in human genome, which is an amazing number. And those are all involved in several different processes, such as cell cycle control, development, nervous system, and other systems and other functions of the body. Other than small interfering RNAs and microRNAs, there are long non-coding RNAs as well. These are found in eukaryotes, just like the other two that we just talked about. These are generally very long, about 200 nucleotides long. They have a 5' cap, and they also have a poly A tail, but they don't have any start or stop codons, so their role is not to make any proteins. It is estimated that they probably interfere or they modify the chromatin structure, and they are involved in some type of gene expression regulation as well. According to some estimates, there may be more than 17,000 of these long non-coding RNA molecules in human genome. And some of these, long non-coding RNA molecules, they function as competing endogenous RNA molecules. So how do they compete? What they do is actually they soak up. They serve more like sponges to soak up the microRNA molecules because of the complementarity that is found between these competing endogenous RNA molecules and these MREs, microRNA response elements, which are found on the 3' UTR region of microRNAs. So those are the ones that are called as mREs. mREs are microRNA response element. So, and these competing endogenous RNA molecules have some complementarity to that region of microRNAs, and that's how actually they depress them. They bind with them and kind of neutralize them. So when they are neutralized, that means they are taken out of the function, and that can probably help the process of gene expression. That probably helps the process of making more types of making more proteins from them. The expression probably is helped. Since they compete, they remove the microRNA molecules, and the role of microRNAs is to reduce the amount of gene expression, and in that way these the cRNA molecules work in the opposite direction. They remove something that decreases the gene expression, so that way gene expression is helped.